welcome everybody to the AFOSR 60th Anniversary Seminar Series. Today we're pleased to have Chad Mirkin, who's the director of the International Institute for Nanotechnology at Northwestern University. Chad was with us actually at our spring review to give us a talk as well, but today he's going to talk about another topic, I understand. So Chad actually is recognized for DiP-Pen Nanolithography, and that's actually the title of his talk, or the topic. Wanted to point out that I think he was just recognized by National Geographic as the #1 of 100 scientific discoveries. So that's recently come out for this work actually in DiP-Pen Nanolithography. He's a member of the President's Council of Advisors for Science and Technology. He's won many awards. I won't go through them. When you introduced him, he kept going through all the different awards that Chad had won. We won't do that again, but if you'd like to see that, you can watch that as well. He's authored over 400 manuscripts, and I think you actually have more patents than manuscripts at this point in time. Comparable? Okay, well, you had that wrong then, so you'll have to pick on him in the future. All right, so he's currently listed as the most cited chemist in the world, with the second highest impact factor and the top most cited nanomedicine researcher in the world. So he's doing a great job based on our AFOSR funding. And we're really happy to have him here, and we appreciate him coming back for a second talk and participating in the 60th anniversary series. So with that, Chad, thank you very much. And
great.
Thanks.
Thank you. Can you hear me? Is it on? Oh, it's not. Okay, so there's no mic up here. Okay, great. All right, well, thanks for the nice introduction and an invitation to come back here and tell you about some of our work going on at Northwestern. I do feel a little bit like Bill Murray in Groundhog Day. The talk will not be the same, the person will be the same. But I do want to go into much deeper detail about an innovation that has grown out of my lab for, I'd say, about 13 years or so. And it started with AFOSR funding. There have been lots of other folks that have significantly contributed along the way. But this is one of those kind of out-of-the-box types of ideas, thinking about doing things very differently from the rest of the world, and actually being able to do something that's really quite unique in the area of nanoscience and nanofabrication. And in fact, the reason I chose it was I think this was selected a while back as one of the AFOSR's significant innovations in a relationship to, maybe it was the 60th anniversary or the look back in terms of the last decade of research, and that's this area of DiP-Pen Nanolithography. But to get started, I wanted to kind of get you to think about something in a way that I think about it, at least. And that is that if you look at these two types of technologies, you can recognize them, I think, almost immediately. The printing press on the left, the modern microfabrication facility on the right. I don't think you have to argue very long to convince people that these two technologies revolutionize the world. On the left, in terms of how we transmit information. They don't have a microphone. This is just for the, I'm not loud enough? I'll go a little louder. On the left, we've got the printing press, which of course revolutionized the way we transfer information and communicate. On the right, of course, the basis for the modern microelectronics and now nanoelectronics industry. Two things, though, that I think are important in terms of observation. The one is that when we talk about printing with respect to electronics, we're extremely good at doing that with solid-state materials. We're not very good at doing that with soft matter, structures that chemists find interesting, material scientists find interesting, and biologists find interesting. Over on the left, we now take for granted that we can have a tool like a desktop printer that allows us to do effectively what very sophisticated printers and printing operations did in the past. And we expect to have that type of capability at the point of use. And so one of the things that we've been looking at, is it possible to one day have the equivalent of that for microelectronics and building nanostructures that are made of biological and chemical-based materials, soft matter-based materials? And that's a pretty tall order. So how would you begin to build a system that would allow you to rapidly fabricate at the point of use any sort of structure you'd like? And so I talked a little bit about this in the last talk, but I want to go into great detail because this is, I think, a pretty incredible story. And there's a tremendous number of kind of scientific and engineering-based innovations that are really critical to make this happen. And it's worth knowing, I think, especially at the Air Force, what the history of that is, given, I think, the importance of it in terms of how people are beginning to use it, what it can ultimately become. And I think if you look at some of the things that happened over the past decade, I think you'll find that there's some pretty neat lessons that we've learned and things that we can build upon to really enable ultimately this idea of creating a desktop fab. So the technology I'm going to refer to is DiP-Pen. In its simplest form, the basic idea here is you take an AFM tip, a cantilever, you coat it with molecules, you bring it in contact with the surface. The first innovation was to recognize that there's a problem in scanning probe microscopy, and that is that when you do an experiment in air, water collects at the point of contact. That's called the capillary effect. That sucks the tip down and then convolutes any measurements you make thereafter. Remember, these types of instruments were designed as imaging tools, not as writing tools. Our idea was to begin to transform this from a reading tool or an imaging tool to a writing tool. And the basic idea was to coat the tip with molecules. Wow, this is terrible. And then bring the tip in contact with the surface. And then when that happens, you could use the water effectively as a carrier solvent to facilitate the movement of molecules from the tip to the substrate. And so as you scan across here, the idea would be that you'd be working with this type of equilibrium process here. In fact, I use this in general chemistry now quite a bit to talk about Le Chatelier's principle, because you're effectively milking the equilibrium towards the substrate. The molecules chemically react with the underlying substrate, and they form a self-limiting type of structure, one molecule thick. And of course, you can make anything with the resolution of an atomic force microscope, and that's a lot higher resolution than many of the tools that we typically associate with the conventional micro and nanoelectronics industry. In addition, of course, the beauty is, in principle, anything that can flow from the tip to the surface and is compatible with that meniscus can be used in a DiP-Pen type of experiment. So at the end of the day, you have an incredibly high resolution technique, 10 nanometer line width, 5 nanometer spatial resolution. Writing and imaging are done with the same tool. That's really critical, and a lot of people don't understand the importance of that, but you always know where you are. So what you have is not only the ability to make tiny structures, you have the ability to overcome one of the biggest limitations in the whole nanofab area, and that is registration. You can place them with incredible registration capabilities because the drift with respect to this type of instrument is less than a nanometer for many minutes of use. And so I can begin to very precisely make structures and place them where I'd like, and that opens up a lot of opportunities, certainly on the scientific side, and I think I'm going to argue towards the end of the talk, even on the fabrication side of things. The other beautiful thing is this is molecule general, substrate general. The critics, of course, would look at this and say, well, this is nice, but in reality, you're only patterning a 90 by 90 micron area. So this is horribly slow, and that's true. That's a reasonable criticism of it. But of course, we're just doing science and publishing nice papers at this point in the development of DiP-Pen and kind of putting this out there as a possibility to move forward. But it kind of set the stage to think about how we could then go from this type of capability to one that would truly be a production tool and enable that concept of a desktop fab. The first thing we had to do was to create a system that many people could use. Because my view is, I think that you have impact by publishing nice papers, but you really have impact when people start using your ideas or concepts or tools. And so there's only so much we could do with this in terms of making it something that many people could use around the world from Evanston. But we started a company called Nano Inc., and the goal of that company was to begin to create tools that would allow you to do nanofabrication through the DiP-Pen type of process for lots of different purposes. But the biggest purpose in the early days was to enable researchers, other researchers, to do nanofabrication and DiP-Pen nanolithography. In fact, one of the early buyers of this tool, I think, was the Wright-Patt Air Force Base, and they began to try to implement and use DiP-Pen and still use it today, as a matter of fact. This now is all over the world. I'll show you that map towards the end of the talk. But the point here is that with a commercial module, an AFM module, a piezo-driven stage, in a controlled humidity chamber, that's important because it turns out that's one of the parameters that allow you to adjust flow rate. You can ratchet up the humidity or decrease it depending upon the types of molecules you're trying to transport. Software that is incredibly sophisticated. I can literally take anything, your picture, an integrated circuit, a gene chip, a protein array, And if I know the diffusion properties of the molecules under a set number of conditions, I can predict ahead of time flow rate and therefore feature size for a given tip substrate contact time. And so not only can you make these types of structures, but you literally now, an undergrad, can program the type of structure they want, and the instrument will tell them at a given resolution when to come back and pick up the results. In fact, it's better if you're not in the room, because vibrations can wreak havoc on some of these types of experiments. Okay, so why is this such an interesting innovation? People have done scanning probe lithography in the past. And this is, I think, one of the big scientific advances in terms of DiP-Pen. It was a recognition that instead of going the way the rest of the semiconductor industry went, which was to deliver energy to a surface to enable patterning, which of course is incredibly powerful. I'm not knocking that. That's a really important concept. Let's deliver materials directly to a surface. Other folks had kind of gone the semiconductor route. They had thought about putting down resist layers, for example. Silicon even, and oxidizing it. Calquate had done anodic oxidation. People had done things called nanoshaving, where they'd put monolayers down and use the tip kind of as a rudder to generate negative types of structures in those resists. The millipede, which a lot of people say, well, don't we have mini-pen types of systems already? The millipede is not good for doing nanofabrication. In fact, one of the first groups we went to see was IBM to figure out whether we could do DiP-Pen on the millipede type of system. And you can't. That's an incredible gross generalization, because the millipede was actually designed to do one thing. For those of you that are aware, this is a 1,000 tip type of scanning probe array. It was designed to burn holes in polymers and then to anneal them. And they wanted to develop it for information storage purposes. Well, we can't use hot tips. We have a hard time using hot tips for most of the DiP-Pen types of applications. And in fact, there's no way to use the millipede and directly do DiP-Pen with it. We looked at it in great detail. And I'll talk about why that is in a little bit, and the importance of some of the observations with respect to materials delivery in a second. I want to show you some of the capabilities, though. If you look at this, these are all structures made by DiP-Pen Nanolithography, kind of in the early days to the last part of or last decade. You can see we've made structures made of silver, gold, electrodes that have really small gaps here, silicon features, sub 50 nanometer features. These are made by depositing molecules onto these types of substrates, using the molecules as etch resists, etching the remaining areas, and you're looking at the ray structures that are left behind. Now, again, a critic would look at this and say, well, I can make this by e-beam. That's not that fantastic. And that's true, but you've got to keep in mind that this is an incredibly simple process. So this idea of going to a desktop fab, you have to enable, but you also have to keep it simple. And that's been kind of one of the mantras of the group. We want to understand the system and how it works. And as we develop it, don't make it so complicated that the only group in the world that uses it is in Evanston. And that actually is what happened with the millipede. To this day, the only group that's ever used the millipede is at IBM. And it is a very sophisticated system, as I said, for building those types of hole-based structures. These are things that you couldn't make by e-beam. They're made out of materials like sol gels, nanoparticles, carbon nanotubes, where we actually create affinity templates that take and adhere the carbon nanotubes from solution and arrange them not just on the substrate, but bend them into unusual conformations. And one of the things that we wanted to look at was how that a strain then affected the properties of these types of systems. So you can kind of think of this as a way of either acting as the tweezers or creating chemical-based tweezers that allow you to pick up nanostructures and place them where you would like, which again was a very important innovation in the early days. You look at these types of systems, again, none of these could be made by e-beam. This is Feynman speech, plenty of room at the bottom, written with molecules. Again, color has no meaning on this scale. This is a false color scale where the white is high friction, the blue is low friction. We're using the instrument to actually record the image. If you took one of these lines and asked, well, what's the level of magnification here? Take a ballpoint pen line, apply the same level of magnification so you can see this. This line would be about a mile wide, or the ballpoint pen line would be about a mile wide. So these are enormous, very, very small miniaturized structures. Combinatorial libraries of small molecules, oligonucleotides, viruses, proteins. We'll talk about what you can do with these in a little bit. All sorts of polymers can be used. And we can take nanostructures and interface them with prefabricated circuitry, for example, made by conventional types of methods like e-beam. And so what we're building now is the capability to print lots of different types of structures, demonstrating that type of generality as we go forward, and then ultimately going to ask what we can do with it. And let's consider that. So let's think about it on the biological side of things. You guys are probably used to thinking about spotted array or combinatorial library types of structures if you followed some of the molecular biology and cell biology studies that have gone on over the last 15 years or so. This is a staple of that field, why it allows you to do lots of experiments simultaneously with relatively small samples. And if you think about the feature size that we're talking about here, with low-resolution DiP-Pen, we can put 50,000 features in a single spot here. With high-resolution DiP-Pen, we can put 100 million. I've got a colleague named Mark Hersom who always likes to trump me. He's got a technique he calls feedback-controlled lithography, where in the UHV he can pluck hydrogens off of silicon surfaces and he can modify the surface. And that really tells you the limit. That's as small as you're ever going to get from a chemical system. But it also kind of shows or emphasizes the sweet spot of DiP-Pen. It's very, very high resolution. It's not the highest resolution of any scanning probe lithography out there, but it is incredibly user-friendly and can be run under almost ambient conditions. And that's really one of the reasons we've seen such a scaling in terms of use of this type of technique and technology. Now, so what does this do for me? From a biological standpoint, the obvious is I can do more experiments per unit area. But to me, that's the least interesting part of this. That might be very useful, because you could use really small sample sizes, for example. But what's really neat about this is that you are beginning to print on the scale of biology itself. And so we can take what used to be a microarray and shrink it down and put it underneath a single cell. And I'll show you an example of that in a little bit. I can take viruses and I can control where they are. And if they're non-spherical, I can even control their orientation. And very recently, we've learned how to take individual protein molecules, actually through some AFSR-funded work, and control where each protein is on a surface. And we think we're going to be able to control orientation as well. And that's going to allow us to look at single molecule protein types of experiments in a fundamentally new way, where we can build lots of these structures and site isolate individual proteins and monitor what's occurring at the single molecule level with a very, very fine level of control. So here's some examples of this. This is a tobacco mosaic virus array here. You can see it's a perpendicular array where we've put down molecules, actually carboxylic acid terminated monolayers, that we put coordinate zinc to, and the zinc coordinates then to the carboxylate-rich surface of the virus. And so the virus adheres to each spot that we've made by DiP-Pen, but we've made, in this case, a perpendicular array, so we're controlling the orientation. Why is that interesting? Well, the virologists want to use this to study things like infectivity. What makes one virus more potent versus others, and what is a seemingly very similar lot of viruses? Are there elements of cooperativity? Do two viruses in a certain type of configuration and interaction with a cell work better than one in terms of, or 2 times 1 in terms of infectivity? All of that has to be looked at from a statistical standpoint and scaled down from the bulk with current methods. But with these types of methods, you literally can program what the cells see at the individual virus particle level. And I think that's pretty exciting. Over here, oligonucleotide arrays. These are the highest density gene chips ever made. Phospholipids are fantastic inks in this case. They flow from they actually do not chemically adhere to the surface, at least the types that we use. We use electrostatics to bind them to the surface. Proteins. And when you see all these, you say, oh, what I have now are a lot of the components that make up the extracellular matrix. So in principle, what we're saying to biologists, if we had this desktop type of capability, you're one day very soon going to be able to program models for the extracellular matrix and use those to do very well, highly controlled biological studies. For example, in understanding how cells bind to surfaces, how they grow, how they move, how they differentiate. I'm going to give you an example of that in a little bit, and ultimately how they die, and look at some of the chemical cues in a highly controlled manner that control a lot of those key processes, which will be, I think, a big boon to the whole field of biology. You can make integrated electronics as well. So we're not making arrays yet of integrated electronics, but we are making masks. And the way we do that is we take a quartz substrate. Usually you have chromium on quartz as your mask. We put down a layer of chromium, a layer of gold, and then we pattern on the gold alkane thiols that define the pattern that we want to make up our photomask, for example. In this case, it's interdigitated electrodes. We then etch the gold and the chromium. We remove the gold through a gold etch and the molecules, and then what we're left with is this type of structure, a simple photomask for interdigitated electrodes. And the point is, this can now be made. We've actually created linear arrays like the one shown here, where you can duplicate the number of pens that are being used, so you can increase the throughput by the number of pens that are used in the array and stitch the structure together. But then you can use these types of photomasks then to literally produce interdigitated electrode devices. And these are some of the devices that have been made, and here we're making measurements on polypearl coated nanotube structures. So that tells you that these are in fact functional devices as well, not just fancy images. Okay, so let me tell you one of the big problems associated with DiP-Pen. So I'm talking about a lot of the strengths. Problems and also potential advantages. One is that if I want to make big structures, in addition to small structures, it's actually pretty difficult to do in a DiP-Pen experiment. Because what happens is you bring the tip down in contact with the surface. Molecules begin to diffuse from the tip to the surface and anchor onto the substrate. And again, if you're working on a small length scale, that's a reasonably fast process. But if I want to make micron or mini micron type structures, it takes a long time for that diffusion process to take place and for you to get stable structures on the surface. The other observation that's important is the following, that unlike in an energy delivery type of tool, if we look at how feature size depends upon contact force in a DiP-Pen experiment, because it's materials delivery and it's really materials diffusion that's important, it's independent of contact force. It doesn't matter how hard I push. Why is that important? Well, that turns out to be a critical observation, because a very talented postdoc in my group, a guy named Sung-Eun Hong, said, you know, what we can do with this is we can build large arrays in principle, and we can overcome one of the biggest problems in this complexity versus capability issue. Remember I said we want to go to big arrays ultimately, but we want to keep it simple. Well, the millipede approach to that in terms of creating these little heated types of structures, they put a little feedback system on each tip, they put heaters on each tip, and now they can regulate the positions of those tips, and they can regulate how hot they are and how cool they are. The problem is that becomes a nightmare from a complexity standpoint. Well, he said because of the fact that we have this situation where the feature size is independent of contact force, in principle what we can do is we can make large arrays like the one shown here. This is the largest array ever made, or actually at the time it was the largest array ever made, 55,000 pens in a square centimeter. So these are all AFM cantilevers, and you can see the high magnification image here with the tip. And here's the problem. When you have an array, whether it's linear, and it's really bad if it's two-dimensional, you can never get all the pens perfectly aligned in one plane. So one's going to hit first. And so if I push and get them all in contact, if I'm delivering energy to the surface, the problem with that is I have a different contact force at each point of contact. But with this type of experiment, in terms of the DiP-Pen transfer of molecule, it doesn't matter. And so what I can do is I can effectively use gravity and bring all the tips in contact. And if the shortest tip is in contact, if I know it's in contact, then I know all the other tips are in contact. So we actually intentionally put in tips that are a little further away so that we know if that's in contact, we've got all the other tips in contact. And in principle, we should then be patterning molecules over, in this case, square centimeter areas. And this was a big milestone. You have to, again, think of the challenge here. We now go from being able to print that 90 by 90 micron area to square centimeter areas. And here, these are all the tips printing, and we're making 88 million features in 20 minutes. Each of these were alkane files deposited on gold. And you're looking at the structure after we've etched it, so you can see the raised features and characterize them by things like electron microscopy. And you can see the uniformity is remarkable. I think it's about as good as you could possibly get with any type of tool, given the roughness of the surface that we're dealing with. And so this was very encouraging and suggested that there might be an opportunity to really scale here and to do something pretty important with this type of scanning probe lithography. And so we were actually almost getting ready to stop at that point. And say, okay, let's call it a win. We've done about as much as we can possibly do. You know, companies should take over and run with this. But then another talented crew of students came in, and one of the things we had been thinking about was the following. These are fantastic examples of taking the single pen DiP-Pen type of experiment and scaling to the point that you can do square centimeter patterning capabilities or have those capabilities. The problem is they still do get pretty complicated. You're still working with a cantilever array. Aficionados don't mind that so much, but the normal user does. So again, if you're thinking about the desktop printer as your as what you're ultimately shooting for, we're still a ways away from that on the micro or nanofab side of things. And so we thought about it and said, is there any way we could go from this type of array to something even simpler? And what we came up with is the following. And I think I've got key advances here. So the first obviously is the materials deposition. The second is this. If you think about a cantilever, it's effectively a spring. And we said, why couldn't we put the spring in the tip? And how would you do that? Well, you could make the tip out of a springy material, an elastomer, for example. And so that opened up this field of what we call polymer pen lithography. And this is fairly recent. This is only, what, 4 years ago. Okay, so all this has happened in the last few years. And then we looked at that and we said, that's kind of nice, but the problem with that is those tips are going to be soft. And so we're going to pay a price in terms of resolution. And we said, if this is a good idea, then why don't we move the spring to a backing layer and put hard tips on it, for example, made of silicon. And then you could maybe have the best of all worlds. And that opened up what we call this hard tip soft spring lithography. And then there are a whole series of other innovations that came out of these types of observations or approaches that I want to share with you that I think are really enabling. And I kind of refer to this as cantilever-free molecular printing or scanning probe lithography. And I think this idea of using a pyramid array when we look back is going to be very, very important because it dramatically simplifies everything that I'm going to be talking about. So this is how you make these structures. Pretty simple. You use ordinary photolithography to make a master. You just make a series of holes in the resist. You then etch the exposed silicon with KOH. It's an anisotropic etchant that naturally gives you these pyramidal pits. You then pour in an elastomer, put a transparent slide over here. That's important. A hard backing that's got to hold this. So you couldn't just use it as a stamp, for example, on a micro-contact printing type of experiment. That wouldn't work. And then you cure this, peel it off, put it into the instrument, the same instrument we use to do atomic force microscopy. Really, any piezo stage is going to work at this point. We don't even need the AFM, as you're going to see in a little bit. And now we're going to use that to control the position of this, and we're going to use these as point sources to deliver molecules to an underlying substrate. Now, the beauty of this, if you're thinking, you say, well, this is kind of a stamp, isn't it? And it is in one form. But I can use this to create a dot matrix pattern of anything I'd like. I don't have to make a new master or a new mask every time I want to make a new type of feature in principle using this particular approach. And so this shows you the scalability. These are some of the actually initial wafers we've made. You can see the size of them. It's 11 million polymer pens in this type of device here. And we simply put that in, use it like a normal AFM cantilever, and bring these down once the array is leveled. We can look through the back of the array, which allows us to tell if it's leveled. I'm going to show you an image of that in a little bit. But this is kind of interesting. If you look, the reproducibility is pretty extraordinary. And in fact, this was the first experiment we did with the 11 million pen array. We put it into the device, bring it down, depositing alkane files on gold. It's octadecane file, I believe, in this case. And then we move it over, make another dot, move it over, make another dot, move it over, make another dot. And you can see the 4 dots duplicated 11 million times. Each dot takes less than a second to make. And we'll talk about resolution in a second. OK, so then the guys come and they show me this result. And they said, do you like this? I said, yeah, that's pretty spectacular. How'd you make that? And they said, oh, we made it with polymer pen. And so you look at this and say, why is it so spectacular? Well, it's spectacular because the quality looks pretty impressive for what I've shown you so far. And this is literally the second experiment that was done. That tells you how easy this is. But what struck me immediately was that these are large features. These are micron scale features. And in fact, the dots used up to make the runner were 600 nanometer dots. And this is just, of course, the Beijing Olympic logo. In the Beijing 2008, with the 5 rings, in this case, were made at 90 nanometer resolution. And see, how did you do that? Because as I told you, in a DiP-Pen experiment to make large features, it takes a long time. And these guys did this very rapidly in a matter of minutes. And they said, oh, there's an interesting observation here. Well, first, I wanted to show you can make any set of structures you'd like. These are integrated circuits. And I don't think there is any other tool out there that could do this, and again, with one tool. And the reason I say that is the pads out here are millimeter scale. The leads are microscopic, and the electrodes in this case, in the middle, are nanoscopic. And so one tool allows you to span multiple length scales very rapidly. Normally, you'd make the bulk of it by microfabrication techniques, and then you would use E-beam to make the fine structure, the structures that have need nanoscopic resolution. Well, this is the reason they could do it. So like a DiP-Pen experiment, feature size, this is edge length, is dependent upon dwell time. But unlike a DiP-Pen experiment, there is a contact force dependence, and that is because these are squishy tips. Now, there's a real lucky part of this, and that is, it turns out, you know, I said, how can you make these structures so uniform? How do you get the array to function like this? Because if they're going to squish, if I have different contact forces, in principle, every tip is going to have a different feature size. But what I showed you, was remarkably uniform. And this is the observation. If you look at this, what you see is that the feature size is a function of relative Z piezo extension. So that's how hard we're pushing in the Z direction. Initially, it's incredibly forgiving. What happens is the base of the array absorbs most of the contact force before any of the tips are deformed. Then you get to a point where now the tips begin to uniformly deform. And in fact, I'm going to show you that's very modelable. We can literally model that and automate this to create a very, very sophisticated system. And so I kind of think of this almost like a gel pad in your shoe. Your sole doesn't deform, but the pad does. But if you kept pushing, eventually your sole would deform. There's more resistance there. And so the architecture here, and remember putting it on this glass slide, is really quite important in terms of enabling these capabilities. This shows you the reversibility. This truly is a soft structure that's springy. If you think about it, as I push harder, I'm taking a deeper and deeper slice of the pyramid. That's why at large contact forces I could make those 600 nanometer features, and at very soft touches I could make those 90 nanometer features. This is pushing lightly, pushing hard, and then going back the other way. You can see it's remarkably reversible. As I said, it's just from simple geometric arguments, it's very modelable. You have to take into account, though, that the polymer has a compression modulus that depends upon contact force. And so that's important in terms of modeling this, because you can't model it all on this one. You have to then throw into your model a change here when you get to a few millinewtons in terms of contact force. And that takes into account the restructuring of the polymer chains within the elastomer as you're applying a larger and larger load. Here's the real thing you should be thinking about for the aficionados thinking about what the problem potentially might be here. Remember I told you that leveling is really important, right? I have this problem where even if it's perfectly level, not all the tips are in contact. But I have this problem. If I bring down the array, one side might hit first. How do I get the whole system perfectly level? And so we began to think about that and in fact model it. This is the issue. You bring down the array and you can see it's intentionally tilted here, and I've got theta and phi. And if we just have a few hundredths of a degree, I mean, you go from tips being in contact to tips being nowhere near the surface. And so that's not going to allow you to build the types of structures that I just showed you. And it's actually remarkable that they got it to work before the innovation that I'm going to talk about now. Okay, so we said it'd be really nice if you just didn't have to have a guy who had a real green thumb to do this, and you could make this something that was almost programmable. And so we began to model when there would be the greatest contact force. And it turns out it's when the array is perfectly level. When all the tips are in contact with the surface, that's where you reach a global maximum in terms of contact force with the surface. And so we said if that's true, then what we could do is we could put an ordinary scale, a balance, underneath the substrate holder, and then just use the balance to weigh, measure the effect of the contact force with the substrate. And then we could tweak with screws that adjust the tilt, theta and phi. And so if you look at this, here we're zeroing in on theta, we adjust, and all of a sudden we see a local maximum in terms of contact force. This is modeling over here, this is real data. This is not the global maximum that we modeled over here. This is what we'll call a local maximum. So we keep that fixed at that point. And then we tweak phi, and we see another local maximum. And then we adjust right around the sweet spot, and you can see the global maximum here. And when you do that, in principle, the array now is perfectly level. Now, you can test that. And you can do that by coating this with molecules and then printing on a gold substrate. And here you can see the perfectly level array. There are 15,000 pyramids in here, 1 square centimeter, leftmost side, middle, and right. You can see the feature size. It's effectively, the variation is as good as you're going to get with any sort of lithography when you take into account roughness of the substrate. There's no variation, significant variation in feature size. Here's why you have to be so good. 1/100 of a degree, right? Look what happens. Small, medium, large. So we're pushing harder on this side, right? And less so as we go across here. Flip it around, large, medium, small. And so if you're off by 1/100 of a degree in terms of theta in this case, that's going to kill your whole experiment if you're trying to make many features of the same size over large areas. But now what I can do is I can literally use this as a way to quantify and figure out when the array is level and when I'm ready to go make these structures. Second thing you'd like to be able to do that a desktop printer does quite well is it uses lots of inks. In our case, inks are not defined by color, they're defined by composition, so we have a much bigger palette to choose from. And many of the structures that we'd like to work with are biological systems. So you can see here, we can take effectively the mold that was used, not effectively, the mold that was used to make the arrays. The tips are nanoscopic in terms of sharpness, about a 60 nanometer radius of curvature. The wells, though, are microscopic in size, and you can hit them with an inkjet printer. So you can fill the wells with anything you'd like that you can transport in a printer. We put dyes on proteins in this case, and even if you get a little splash, it doesn't matter because these are quite a bit away from one another. And now if I get 2 pens in, I have all 15,000 pens in. And so when I do this, I can coat the mini tips with different types of molecules. I can now bring the array down onto the substrate, and I can begin to pattern. And each of these are different protein structures. You can see by the different dye labels. And here you can see we can build an array that varies in feature size by controlling contact force. And so if you look at this and ask, what the limits are, about 65 nanometers in terms of feature size, highest high-resolution structures I think that can be made by any technique, up to 365 nanometers pretty routinely. And so we've been working out all the chemistries in biology to actually use these types of structures as well for a variety of different purposes. So then you start thinking. You say, okay, this is good. We spent a lot of time figuring out how to make these arrays level. What could we do with a system that was intentionally misleveled? And it turns out you can do some pretty interesting things. So if you think about this, if you took and now adjusted theta and phi intentionally, you could begin to build a situation like this, where parts of the array will be lightly touching and other parts of the array will be kind of smashing into the surface. And if you think about that from a printing standpoint, what that allows me to do is to build gradients of features. And so now I can open up what I like to refer to as the field of nano-combinatorics. And if I use biological structures, I now can begin to build large libraries. You think of 11 million pens in a half a second. I can transfer a feature. I can make a library very, very rapidly. And that has a lot of different reactions that can take place. And then I have to have a way of reading out the results ultimately. And so the question is, what would you like to probe? And I think if you think about it, there are tons of things we can probe. This is not just biology. We're talking about plasmonics. We're talking about catalysis. There are all sorts of interesting things we can do with this approach if it's designed to work extremely well. So here's an example. This is not just conceptual. These are fibronectin arrays. And a woman named Louise Guillaume, who's since moved on to Stanford to do a postdoc, just finished this work. It actually just came out in PNAS. And the reason she wanted to print fibronectin arrays is fibronectin is an adhesion protein. And what we thought was what we could do is begin to build libraries of different size structures and maybe different compositions, and then use cells as a readout, because they're microscopic. I can visualize them with ordinary types of microscopies. And I can ask how different feature sizes and feature compositions and feature shapes and interfeature distances affect things like cell adhesion, cell growth, cell motility, cell death, and in the case of a stem cell, a mesenchymal stem cell, differentiation. And why might you think that would be important? Well, it turns out if you look at mesenchymal stem cell differentiation, you look at the different types of structures that these things can form, the different lineages that result from this, bone cells, for example, being one of them, nerve cells being another, fat cells being a third. If you look at the way they interact with surfaces, oftentimes they form very different sized adhesions. And so one hypothesis would be that if I controlled the contact area of the adhesions, I might see a preference in terms of differentiation. Well, to make a long story short, she goes through and she does a tilted array type of experiment, and she screens by looking through a microscope what the cells do. And she says, oh, I see these preferentially form in this part of the array osteocytes. I'm going to go study that in more detail. She doesn't conclude that she has osteocytes at that point. What she does is she then levels the array and duplicates that particular set of features in mass so that you can get statistically significant data. And then she goes in and measures biological markers for osteocyte formation and proves that you are in fact controlling differentiation. What's neat from this study is that she found that 300 nanometer features will selectively trigger the formation of osteocytes better than the known chemical cocktails for controlling differentiation, simply by controlling focal adhesion side. If you go to 1 micron scale features, you don't get that type of control. Now, why is that important? That opens up a whole new toolkit now to study these types of processes. And we would have been happy just to see some slight preference here, a slight preference, because we hadn't even used all the different chemical cues. We're only using 1 protein in this case. And so when you put the multiple types of capabilities here, I think, again, it's going to create, kind of think of it as a way of rapidly figuring out what types of models and cues can be used to control really important cellular biology types of experiments. And so we're putting that out there now as kind of a challenge to that community to begin to run with this. Okay, remember I said that the second innovation you could go with is now to move the spring from the tips to the backing layer. Why do you want to do that? Well, remember we go to 65 nanometers. With DiP-Pen we can go down to about 10 nanometers. How would you do that? Well, you could fabricate silicon or silicon nitride tips on an elastomeric pad with a transparent backing. Here you can see you can do that. These are really kind of interesting. If you look at this, it looks completely transparent. Most of this is polymer. These are silicon nitride tips. You can see the uniformity is pretty remarkable here, and this is the big advantage. Now I have these hard tips resting on these soft springs, or the soft spring layer. I can make these very, very sharp. We think we can take them down to 10 nanometers of radius of curvature. But now when I use this type of system, I literally can look through the back of the array, and you guys can see this, looking through an optical microscope, I hope. There we go, good. When the array makes contact, you can see it deform. So you now literally can use the naked eye to follow when these are making contact with the surface and rapidly align them just by inspection. And when you do that, you can see as the tips make contact, if only one side of the array was in contact, you'd see it deforming, whereas the other would not, and vice versa. You start characterizing this. This is more like a DiP-Pen type of experiment. Feature size is independent of contact force, right? Because the molecules are going to flow. But we can control feature size by dwell time. Now you can begin to build very sophisticated structures. There's sub 50 nanometer, 42 nanometer features here. This is the Eye of Odin off a US $1 bill. I think duplicated 15,000 times. Every structure is where it's supposed to be. And you can see the high resolution 42 nanometer dots here used to make up the Latin writing and also the pyramid. But you can begin to do a lot of neat things with this. So one of the things we were always trying to push on are the limitations of the technique. And so I keep talking about 10 nanometers being the high resolution DiP-Pen type of experiment. That's if you're really, really good, and that's if you're working on really, really flat substrates. It'd be nice if you could, first of all, do this routinely, and also break the 10 nanometer limit. I think there'd be a lot of neat things you could do from a physical sciences standpoint and material standpoint if you could do that, and do that with ease. There's no way it's going to happen that I can see with pure scanning probe types of experiments. But here we took a page out of what's called the block copolymer lithography area, where we said maybe what we could do is create a polymer that was designed to flow. So we have a polyethylene oxide over here, and poly-2-vinyl pyridine as a block that was designed to pick up something that was going to be used as a synthon to make a resulting structure, let's say a metal ion. And so if we take this and put this, for example, in water, these will form micelles. We can load them up with metal ions because the metal will bind to the pyridine in this case. And now we can put these on tips, either a cantilever or the polymer pen arrays that I showed you, or the hard tip on soft backing arrays, pattern on a substrate, And what this does for us, it creates features, polymer features, where the volume of the feature precisely controls the number of atoms, metal atoms, that can be used in a subsequent synthetic step. And so if all things work well, the idea is we're going to reduce with a plasma. Gold will go to gold 0, begin to cluster, and form nanocrystals where the size of the crystal depends upon the polymer volume. Here you can see the uniformity is pretty remarkable. And what's important here is not just the diameter of the features, but look at the height. If that doesn't work, then none of this works. So being able to control all these parameters is what's critical. And this is then the experiment. I think you can see, at least up here you can see, but each one of these are dots. You can see the dots? Very hard, I think up in the front you can see these, but if I magnify these, you can see they are literally gold single crystals. You see the lattice fringes, they're gorgeous. Every atom in that polymer feature gets reduced and comes together to form a little single crystal nanoparticle. And so in principle, what that means is you have a way to control particle size. So here is the polymer feature size, and here you can see the resulting particle size. And so now I can dial in the size of particle I want, and I can control the placement of it using these high-resolution scanning probe techniques. And here you can just see the type of positional control. This is made up of nothing but gold single crystals. Okay, the Northwestern logo, because I can now positionally encode in the DiP-Pen type of experiment. I need the lights off to see the other part. This is it if you look at it carefully. Say, okay, I'd like to now break 10 nanometers. How do I do that? Well, I work with a 50 nanometer polymer feature. I reduce. Look at the variation in particle size. I don't think you could make these even with the best known solution synthetic methods. So this is really a very fine control over particle size, because I can't use up obviously more gold atoms than I have, and the polymer feature defines how many I actually have. And here you can see it's plus or minus a tenth of a nanometer. And now we're going to be able to look at this in real time by cooling it down. And I want to just make a point here. If you look at this, this is low temperature electron microscopy images, and you're going to see there are particles over here. We're watching the particle grow over time. It's going to eat this particle. And you're effectively watching coalescence and Ostwald ripening occur by fixing the size of the reaction vessel, in this case, the polymer feature, and then cooling it down, reducing and allowing the system to basically coalesce. And you can then take this apart and begin to see images like this, where you can see at various stages, you can see all the different sized particles here. Eventually you get down to one big particle here. I think it's maybe better on the next one. Yeah, it is. And here you can see little tiny dots over here. Those are small particles that have not been consumed yet. But what's really neat about this is it gives you a way. I teach general chemistry, and we always talk about Ostwald ripening. Nobody ever shows you that. Well, now you can actually watch it. And in fact, you can differentiate it from coalescence. And it turns out that coalescence is observed for particles closer than 5 nanometers, whereas you get ripening if they're further away. By that I mean, in one case, particles are fusing with one another, and they're kind of consuming those that way. In the other case, they're remaining fixed, and atoms from this are going to atoms from this, and this guy's slowly dissolving while this guy's growing. And you can tease out from these types of experiments that the critical distance is 5 nanometers. So there's the other, in terms of fabrication, there's an incredible scientific play here, the ability to look at these types of processes in a highly controlled way and get information that would be difficult to get almost by any other type of technique. Last thing I'll show you then is then moving this to the energy delivery side of things as well. It turns out we're not only about materials delivery, because when we started making these polymer pen arrays, what we realized was that you can take these types of structures, you can put an opaque material on them, like gold. Remember, these are transparent, I showed you that. And you then can bring these down onto a sticky substrate, something like polymethyl methacrylate. And if everything's perfectly level, the leveling's critical here, I can pull off the gold at the end of the tips. And what that allows me to create are effectively 15,000 light pipes, where I can use the pyramids to channel light delivered from the backside through these tiny apertures. And the beauty of this is I can now work in the near field. And so I can begin to, in principle, use this to develop photoactive substrates underneath with nanometer scale resolution. And so this just shows you some of the apertures that we make, some crudely by the technique I showed here, others where we literally drill holes with ion beams. A lot of this in terms of making it work even better and better is going to be about controlling the size of these apertures. But here you can see we can put a photoresist on a substrate, let's say silicon, and we can develop that and then rinse away the undeveloped area and then etch the substrate so that you can see the results of the photolithography in this case. Again, the beauty of this is I can work down to about 90 nanometers right now. We think we're going to get down to 50 pretty soon. This is the Chicago skyline where we're now making dots and moving the array around, duplicating it 15,000 times. And again, it kind of shows you the precision and the resolution of the technique in a really kind of very crude type of format. But I think this is really one of the exciting areas, both in the chemistry, biology, and also material science and fabrication side of things. One of the things that we haven't solved yet is the ability to independently actuate and control which pens are working at a given time. So far, what I've talked primarily about are duplication tools. In this case, because the backings are microscopic, I can address each of those with light, and I can use a variety of techniques to put shutters on here and control when light is passing through them. So in principle, I can begin to create very large arrays with thousands, if not millions, of pens, where in parallel I can develop and in effect photochemical processes at the tip with nanometer scale resolution. And that would be extremely exciting, I think, in terms of really making this a very simple and very diverse type of capability for enabling that concept of desktop nanofabrication. And so this is just an example where we're putting physical masks over these. We're in the process of trying to build different types of systems and test which ones are good for turning the different pens on and off. Okay, so this is kind of where we are now. So if you think about this, this has been about, what, 13 years or so, going from an initial idea, thinking about using the scanning probe system as a pen rather than a pin, and as an imaging type of system. I kind of look at the most recent developments as a merger of many of the concepts from DiP-Pen and those of contact printing from guys like George Whitesides. In fact, many of these types of techniques take all the strengths of these two techniques, the high resolution, the registration, the high throughput up here, the simplicity, and combine them and get rid of most of their weaknesses. This is not a very high resolution technique. Every time you want to make a different type of structure, you have to make a new master. This is a very high resolution technique, but I can never have the throughput of something like this up here. But when I get into these pyramid arrays, you get into a system that is incredibly scalable, remarkably flexible in terms of the types of materials that you can work with. You can still get very, very high resolution, as I've shown you, and you can combine with things like scanning probe lithography and beam pen lithography to create a materials versatility and access to length scale that I think is almost unchallenged with any other set of techniques that are out there. And so, you know, thinking forward, I do believe we are getting to the stage now where we can really think of these as routine types of production tools. And one of the things we're pushing over the next decade is beginning to use them to really make impacts in areas like biology, photonics and plasmonics, and catalysis, where we can take advantage of this type of structure control to begin to set up experiments that could not, that would provide information that you cannot get from just statistical analyses of bulk samples of these types of materials. And I've given you a few examples of that. But I want to finish by just kind of showing you this. This is an old map. I can't keep it up to date, but I guess this is from about 2.5 years ago. I was given a couple of talks in China. And you remember I said that we start, a lot of people ask me, why do you start companies? And one thing is, you know, I like to see our technology commercialized. But the other thing is, I said, you know, I started by saying impact is measured by those that use it. If we didn't do the companies, if we didn't do the translational part of what we do, if we didn't keep pushing and developing these types of techniques, there'd only be one red dot on this map, maybe a couple, for a couple of aficionados pick it up. But when you begin to build these systems where other people can get them in their hands and begin to work with them, you can have much larger impact. And today there are over 200 different laboratories, I think in 23 different countries now, that utilize some version of DiP-Pen or polymer pen type of lithography. And I think this is going to continue to grow, especially as the systems get more scalable And as I said, we keep them very simple and usable for lots of folks both within and outside of the scanning probe community. And then you guys have already seen this, so there was perfect timing for this. So I was telling Huey, I said, we got to pick something that's really Air Force relevant, and he said, we'll do DiP-Pen, do the whole story of DiP-Pen. And literally last week, they came up with this article, and it couldn't have been better timing for this talk, because they picked this as, we like to say the #1, but Hugh and I were talking about, they actually break it up into different categories, and we don't know if they're ranking them or not, because they have things like penicillin, little things like penicillin in there. Evolution, the wheel was in there, the internet was in there. They were good enough to include nanolithography and DiP-Pen nanolithography in particular. And I think that's kind of looking forward, obviously, in terms of impact. And then I want to acknowledge the folks that did the work. This is my group. Many people from over the last 12 years or so. We actually had a reunion in Evanston. Lots of key people. I mean, one of the great things, and I've been really lucky not only to have great grant support from the Air Force and much of the military over the years, but we've had these fellowships to graduate students that have played a very big role in kids being able to join my lab and work in teams. This is oftentimes, it's not just a few people, it's many people that bring together lots of different components of chemistry, biology, material science, medicine, and engineering, and enable these types of capabilities. And without them, we would not be very far. And then these are the folks that have contributed from other labs. Chang Liu, who used to be at Illinois, we moved him to Northwestern. He was a microfab person, ended up helping us out a lot in terms of the early cantilever arrays. Harold Fuchs and Stephen Lenhart on the biological material side of things are from Germany. Mark Ratner and George Schatz were really important in terms of modeling. I didn't put a lot of those in, but a lot of the computational modeling. We have fantastic theoreticians at Northwestern, and they take a very active interest in a lot of the experiments that are going on and are really important in providing the modeling understanding of things like transport in the context of DiP-Pen. Morley Stone and Rajesh Naik have been involved in the early days of DiP-Pen and now using these types of techniques to make all sorts of interesting biological systems, but also ways of pinning down nanostructures. And I talked about some other structures in my last talk made by on-wire lithography, where we're beginning to use these capabilities and merge them so that we can take devices made in solution and begin to arrange them on surfaces using molecule templates created by DiP-Pen and related techniques to control that type of alignment and registration. Joe Fragala was one of the engineers at the company. He was involved in making the first 55,000 pen array that I showed you. And then, of course, the funding provided by you guys and many other groups along the way. But the Air Force was clearly first in this case at the forefront through a parent grant that I ultimately had with Hugh. I'll stop there, and I'd be happy to answer any questions if there are any. Thanks.
So do we have any questions for Chad?
Do you collaborate with Rogers of the University of Illinois, Scott Rogers?
No, John Rogers.
John Rogers.
Yeah, we do a lot.
It would make sense.
He's kind of working on the micro side, right? He has a lot of scales, and we work on the nano, and there have been a lot of cases for mergers, and we unfortunately were unsuccessful. We were finalists in a big one of these STC competitions, and that would have really launched a big effort between the two labs. But he's been very active in a lot of these types of projects and developing lithographic techniques in general in the Illinois area, so he's a natural partner in that case.
Mike, I'm going to give you the mic.
Beautiful talk, Chad. Have you started, you alluded to exploring some of the plasmonic effects that you can get with these gold hero tips?
Yeah, well, one of the things that we want to do is we want to learn not just the tips. So you're talking about the tips. So a lot of people have brought that up. So you could think about using those tips as local enhancers too, right? There are all sorts of neat SIRS types of experiments you could do where you could use these not only as fabricators but readers, or both. We have not done that yet. That's kind of in the back of our mind. We're trying to kind of take the low-hanging fruit and work our way up through that. What I really want to do is move to independent actuation, because I think independent actuation in this case is very doable without making the system horribly complicated. And if you do that, I think people use this everywhere, not just for fabricating things by lithography, synthesizing things, right? If you think about DNA synthesis and peptide synthesis, many of those types of reactions are enabled by photo cross-linkers or decouplers. And so you can use the pyramids in that case to control that type of chemistry and mass over large areas. And the plasmonic side, we're kind of do the opposite. What we want to learn how to do is to build gradients of features with the polymer pen types of experiments, and then control the formation of plasmonically active nanostructures where we can look on mass at many of these structures simultaneously and begin to tease out structure-function relationships where we're not just looking at 10 or 20 of these, but literally millions of them, and looking at every parameter that you could possibly dream of: feature size, feature shape, inner feature distance, and see if we can find structures that do things that haven't been discovered thus far.
I wondered if you could even photo bleach or photo damage the tip to make holes.
Photo damage.
You know where you were making holes in those nanotips?
Oh.
You get very high fields there and trying to...
That's a good question. That would be, so I mentioned this in the talk. I think that's a clever idea. I have not thought of that yet. The people that figure out how to make those apertures really, really tiny and very, very uniform are going to enable this in I think, very, very grand fashion. Because the beauty of this is that there's this zone plate lithography that what's the guy, Hank Smith has done, which is spectacular. But they're limited right at about 200 nanometers. I think it's 190 nanometers in terms of resolution, because they can't work in the near field. Here you can work in the near field. Now, we're not that much below it. We're actually, I think, at 110 or so. But in principle, we can go a lot lower. But a lot of it's going to depend upon being able to make these tiny apertures, and if we want to have them independently addressable, making them incredibly uniform over large areas. And that's going to take some clever protocols, and it might be that type of approach where you kind of focus in a plasmonic way to damage the tip. So it's worth taking a look at.
I was intrigued by your slide where I think you used the Greek letter phi for an angle or two angles. You mentioned that it was off by just a little bit either way. Basically didn't work, and you had this fairly narrow peak in the middle.
Right. So that's where you've got it perfectly level, and all the tips are in contact with the surface, and you're using the scale to measure the contact force. And so it turns out if you're like this and you've got, let's say, 80% of the tips in contact and 20% out of contact, you're somewhere over here on the other side of the peak. But as you adjust and get to this point, there's a very sharp rise in terms of contact force, which can be extrapolated from the weight measurement.
And did you find that peak simply by running a lot of experiments and saying?
Well, that's what I'm saying. Yeah. So we have tilt screws. And so you set this up with a scale, exactly, and you bring it down, you make a measurement. Now you adjust. And you say, are we flat? Or are we getting to a point where it's rising? And in the initial stages, when we're just tilting and changing one screw, we get a local kind of rise, and we say, oh, we're pretty good in that parameter. Now let's keep that fixed and adjust the other tilt screw. And that's where we got the second, what I called local maximum. And then you go back, well, and then you kind of tweak right around there, and that's where you get the global maximum, and that's where everything is locked in. It's really, really kind of neat. I mean, it's the beauty of the elegance is a simplicity in that case, because you can imagine that if a company were to run with this, they could come up with a pretty slick system where the scale is obviously transparent to the user, and it's just doing it all, from a purely automated standpoint. It just keeps adjusting mechanically until you get the array perfectly level, and you get that global maximum, and it says stop.
So I was just wondering, clearly this is a good overview of the convergence of nano and biology and chemistry and materials. So what about informatics? Have you thought about, especially when you get to individual actuation, about data storage?
Well, absolutely. Well, that's going to be an important part. And in fact, with the combinatorial libraries, that's a huge part of it. So figure you get all this data. What do you do with it too? How do you decipher it all? So there's a big info component to that. And that's really one of the challenges going forward in terms of using these types of libraries. Are you talking about that? Or are you
I'm talking about actually as you build the structures, designing in the structures, the ability to do informatics where you can actually do computing on these
high resolution
structures, especially when you have individual actuation. So you could do information storage in these chemistry structures, or you could actually.
Yeah, I mean, that's kind of a little bit like the millipede idea, right? Because they wanted to create an high-density information storage system just by binary, there is a hole, there isn't a hole. Here, there is a component of that. So the company that commercializes Nano Inc., one of their big products is actually in the pharma industry, and it's for brand protection. And they store information and hide information based upon the size of these structures, the physical pattern, but also the chemical composition of the pattern. If you think about it, you've got multiple layers of information in these systems, and it's hidden by virtue of size. So they can build all sorts of dummy structures that are there to take the, quote, counterfeit or down the wrong path, but they only know where the actual structure is that tells them this is an authentic, for example, pharmaceutical. And then they have all sorts of fancy ways of reading these. So that's kind of a step in that direction, but you could take that to the next level if you really move to true computation.
Okay. Any other questions for Chad? Oh, go ahead.
I guess I should use this thing, since it doesn't go out otherwise. So in your cell differentiation, have you done any differentiation of hematopoietic stem cells?
No. Not yet.
I would think that would be interesting.
This is brand new. I mean, this
We
literally.
already know that surface structures will cause a differentiation into red blood cells or platelets.
No, it's definitely new. This paper literally just came out last week in PNAS. And as I said, our main point was not to say, hey, you can do differentiation. That is going to be a very important point, but there are so many things that we can in principle do with this. What we wanted to do is make the point that this tilted array experiment and this technique is a very powerful tool for chemists and biologists and material scientists to exploit in building these libraries that allow you to study. You know, you pick your favorite differentiation event, you pick your favorite adhesion event. In the case of catalysis, you take scanning probe block polymer lithography, you make an array of 11 million particles that vary in size, composition, and interfeature distance. All of those can have significant effects in terms of the catalytic properties. So there's a company, UOP, for example, oil company, very interested in building these libraries to understand different aspects of catalysis in mass and on the nanoscale where it counts.
Okay, before we thank you for the last time, we give a token of appreciation.
Great.
It's a little AFOSR coin. It's actually a 60th anniversary coin. So there's only a few hundred of these around, and we give them to every speaker. Appreciate your
Thank you much. I appreciate
participation.
it.
Thank you very much. And we can thank Chad one more time.